My name is Charles Ducker, and I'm the Senior Director of Analytical Services for North America. I oversee the 2 analytical services labs that operate in North America. Some of the biggest challenges for myself and my team at NAMSA is to meet the regulatory agency's expectations when there isn't always guidance documents out there speaking to their expectations. Sponsors conduct chemical characterization studies in order to show that their devices are safe. Typically, chemical characterization is the first part of understanding the biocompatibility of their device. The data that we generate is used to determine whether any of those compounds are of toxicological concern. This is done by a board-certified toxicologist, and they will do literature searches on the compounds, use the available data to determine whether any of those compounds are a risk. It's extremely important for our sponsors to provide information about the materials of construction, the manufacturing process for the device, because all of that information can help the analysts in the lab identify the compounds that we are extracting from those devices. Extractables testing is not required for every medical device, although if it's not performed, then the sponsor needs to show in some other means that the device is safe. And when we're presenting data to a regulatory agency, we have 3 categories: confirmed, confident, and tentative identifications. The regulatory agencies in the current situation don't like to see tentative identifications. So that extra information that the client is providing about the materials of construction, the manufacturing process, all lead to more confident identifications, which is the goal for our studies. The patient population is important in determining the analytical evaluation threshold and what threshold of toxicological concern. So these are concepts and ideas that are used to set limits for what we are detecting, how low we need to go in the analytical techniques that we're using to identify compounds. When we are performing chemical characterization studies, we often need to choose from a list of multiple types of solvents in order to get one that doesn't degrade the device, because the most important part of the extraction process is to keep the device intact and not degrade it. So there are a couple of areas that can impact the study. One which we've spoken about, which is the solvent compatibility, and the other is the number of compounds which we need to identify when we have a lot of degradation or the solvents are not compatible with the device. This can dramatically slow the turnaround time, because often it leads to a large number of peaks in the chromatography that we need to identify. And when we're trying to identify polymer breakdown products, those are very difficult because those are typically not in standard databases or haven't been well characterized in other published literature. One of the things that we're doing to improve the client experience at NAMSA is we're deconvoluting some polymers, some plastic polymers, so that we can better identify polymer breakdown products. This is an area where when we do see breakdown from the extraction, it often leads to unknowns or poorly identified compounds, and so this activity will help us better identify those breakdown products and provide more confident IDs when we're looking at devices. It is very beneficial to have a toxicologist associated with the program. Our analytical laboratories work very closely with our internal toxicologists. They have a good understanding of the chemistry and what we're doing, and if we are working with internal toxicologists and then they're going to in turn do that toxicological risk assessment, they already have a good idea of what we've been identifying, what classes of compounds they need to look at, how many compounds they need to assess. So that can dramatically shorten that amount of time that it takes to do the risk assessment. Sponsors can benefit greatly by working with NAMSA on their chemical characterization programs because typically not only are they doing chemical characterization, but they're doing biocompatibility, tox risk assessment, biological safety plans. All of these things NAMSA does very well, and we can do them all in one place.